Welcome. This presentation provides a critical appraisal of the TAK-003 tetravalent dengue vaccine based on the results from its 4.5-year phase 3 trial. As you can see on the screen, this was a large, randomized, double-blind, and placebo-controlled study published in the Lancet Global Health. My name is Anupam Singh, and I will be guiding you through an in-depth analysis of this important research. The goal is to carefully evaluate the trial's methods, results, and implications for public health and clinical practice in regions where dengue is endemic. We will begin by looking at the overall verdict first to frame our detailed discussion. Before we dive into the specifics, let's start with the overall interpretation. As summarized on this slide, the trial provides strong evidence that the TAK-003 vaccine is effective against symptomatic and hospitalized dengue in children and adolescents in endemic areas. The risk of bias for this main outcome is low. However, the story isn't perfectly clean for all serotypes. The evidence is weaker for protecting dengue naive participants against DENV 3 and DENV 4. Furthermore, some concerns exist regarding the hospitalization and severe disease endpoints due to variations in clinical practice. The bottom line is clear. We have a useful vaccine signal, but with some remaining uncertainty, particularly for seronegatives. Now, let's frame the specific clinical question the trial was designed to address. This table breaks down the trial's design using the PICO framework. The population consisted of healthy children and adolescents aged 4 to 16 in endemic countries, which is highly relevant for pediatric public health. The intervention was 2 subcutaneous doses of the TAK-003 vaccine, a clear and reproducible schedule. This was compared against a saline placebo, which is appropriate for assessing efficacy. The main outcomes were virologically confirmed symptomatic dengue and hospitalized dengue. As you can see at the bottom, the trial successfully answered whether the vaccine reduces dengue over several years. However, it did not fully answer if it protects dengue-naive individuals equally against all 4 serotypes, especially DENV3 and DENV4. Understanding why these distinctions are critical requires a look at the challenges of dengue vaccine development. Dengue vaccine development is exceptionally difficult for several reasons, highlighted by the numbers at the top of the slide. There are 4 distinct DNENV serotypes requiring balanced protection and 2 key safety contexts based on whether a person is seropositive or seronegative. Long-term follow-up over many years is needed to detect any waning protection or potential late harm. As the clinical background points explain, prior dengue exposure changes the risk-benefit calculation, making seronegative safety a central question. Our appraisal will focus on key questions like whether randomization was successful, if outcomes were meaningful, and how the data can inform local vaccination policy. Let's begin by examining the study's design more closely. This slide provides an at-a-glance summary of the multi-country trial's design. It was a phase 3 randomized, double-blind, placebo-controlled study conducted at 26 sites across 8 countries. The population included over 20,000 healthy participants aged 4 to 16. As you can see under allocation, participants were assigned in a 2-to-1 ratio to receive either the TAK-003 vaccine or a placebo. The follow-up was rigorous, with weekly contacts to identify febrile illness or RT-PCR testing. The analysis primarily used a Cox proportional hazards model. The red bar at the bottom summarizes it well. This was a strong design for assessing overall vaccine efficacy, but interpreting the exploratory subgroup results requires caution. Let's now look at the characteristics of who was studied. Here we see a table of the participants' baseline characteristics, which confirms the groups were well balanced. As the key points on the right highlight, the mean age was 9.6 years, with roughly equal numbers of males and females. The participants were drawn from both Asia and Latin America. Critically, about 27.6% of participants were seronegative at baseline, meaning they had no prior dengue exposure. This is a crucial subgroup for assessing safety and efficacy. The table shows that randomization successfully created comparable groups in both the safety set and the protocol set, giving us confidence in the trial's internal validity. Next, we will examine how well these participants were retained throughout the long study period. This consort flow diagram illustrates participant progression through the trial. Over 23,000 individuals were screened to randomize just over 20,000, following the arms for both the TAK 003 and placebo groups. You can see the numbers for those who received at least one dose and those who completed the study. The appraisal note at the bottom highlights the excellent retention with a 91% completion rate over 4.5 years. Importantly, the discontinuation rates were nearly identical between the two arms at 9.0% for the vaccine group and 9.1% for the placebo group. This high follow-up rate minimizes the risk of attrition bias. Now, let's review the specifics of the intervention and comparator. The intervention TAK 003 is a live attenuated tetravalent dengue vaccine built on a DENV2 backbone administered as two 0.5 milliliter subcutaneous doses 3 months apart. The comparator was a simple 0.5 milliliter saline injection, which ensured that participants, investigators, and sponsors remain blinded to the treatment allocation, as noted under. Why this is useful: this clear schedule and placebo comparison allow for a direct and unbiased estimation of efficacy. However, as the section "What is still unknown" points out, this trial does not tell us about the effects of booster doses or the vaccine's performance in populations outside the trial's scope, such as pregnant individuals. Next, we'll look at how the outcomes were measured. The trial's strength was its objective outcome, ascertainment. The primary clinical signal was virologically confirmed dengue, or BCD, which required both a febrile illness and a positive serotype-specific RT-PCR test. For safety, serious adverse events were monitored throughout the study. The numbers here are striking. Out of nearly 28,000 febrile illnesses reported, almost 98% were tested. This active surveillance led to the detection of 1,007 VCD cases, of which 188 were hospitalized. As the appraisal note indicates, RT-PCR confirmation is objective and specific, but the clinical outcome of hospitalization is more dependent on local admission practices. Let's now turn to the statistical methods used to analyze these outcomes. The statistical analysis was based on a sound, pre-specified framework. As you can see, under the core method, vaccine efficacy was calculated from the hazard ratio derived from a Cox model, which was adjusted for age and stratified by region. The primary endpoint was powered to show efficacy at 1 year. However, several limitations of inference are noted on the right. Most of the 4.5-year subgroup analyses were exploratory, with limited control for multiple comparisons. This means that after stratifying by serotype and serostatus, the event counts became small, reducing statistical power. Our critical appraisal judgment, therefore, is that the main overall results are credible, while the fine-grained subgroup claims must be interpreted with more caution. With this in mind, let's review the overall efficacy findings. This slide presents the headline efficacy results after approximately 57 months of follow-up. The vaccine demonstrated 61.2% efficacy against any virologically confirmed dengue. Even more impressively, it showed 84.1% efficacy against hospitalized dengue, a more clinically severe outcome. Crucially, there were 0 deaths judged by investigators to be related to the vaccine. The bar chart below visualizes these findings, showing that efficacy against hospitalization was stronger than against any symptomatic VCD. This is true for both the overall population and the seronegative subgroup, suggesting the vaccine has a significant clinical impact by modifying disease severity. Let's now examine a forest plot to see these results in more detail. This forest plot breaks down the efficacy against overall symptomatic dengue. The top line shows the overall result of 61.2%. Below that, you can see that for seropositive participants, the efficacy was a consistent 64.2%. The most important part of this plot is the data for seronegative participants. As the note at the bottom explains, there was clear protection against DENV1 and DENV2. However, for DENV3, the point estimate is negative, suggesting no protection. For DENV4, the confidence interval is extremely wide, indicating the data is too sparse to draw a firm conclusion. This highlights the critical differences in protection depending on serotype and prior exposure. Let's organize these findings by baseline serostatus. This table directly compares vaccine efficacy between seropositive and seronegative participants. For the broad outcomes of VCD and hospitalized VCD, the vaccine showed a clinically strong effect in both groups. The key distinction emerges when we look at specific serotypes in the seronegative group. As you can see for DENV 3VCD, the efficacy was negative 15.5%, indicating no protection. For DENV 4VCD, the data was too sparse to make a reliable inference. The key clinical takeaway here is that while the vaccine reduces overall disease in dengue-naive individuals, it does not do so evenly across all serotypes. The next slide provides a visual representation of this over time. These 4 graphs illustrate the cumulative incidence of dengue by serotype over the full 4.5-year follow-up. In each graph, the lower lines represent the vaccine groups. For seropositive participants, the TAK 003 lines are clearly and consistently below the placebo lines for all 4 serotypes, demonstrating broad protection. The story is different for seronegative participants. While you can see separation for DENV1 and DENV2, the lines for DENV3 are nearly overlapping, confirming the lack of observed efficacy. As the appraisal note suggests, this isn't a trial failure but a precision problem driven by the natural variation of circulating dengue serotypes. Let's now shift our focus to the more severe outcomes. This forest plot details the efficacy against hospitalized dengue. The overall efficacy was a very high 84.1%, which is a strong and clinically important signal. As the interpretation note states, high efficacy was seen in both seropositive and seronegative groups overall. However, a critical point of caution is needed. If you look at the DENV 3 results for the seronegative group, there was a numerical imbalance, with more hospitalized cases in the TAK 003 arm than the placebo arm, although the numbers are very small. Excluding data from Sri Lanka, which had high admission rates, reduced some of this imbalance, but the data remains sparse. Now, let's consider how long this protection lasted. These graphs demonstrate the durability of the vaccine's benefit into the 4th year of follow-up. The top graph shows the cumulative incidence of any confirmed dengue, while the bottom shows hospitalized dengue. In both, the continued separation of the curves indicates that protection persisted well beyond any initial short-term effects. The numbers on the right are impressive. During year 4, VCD efficacy was 62.8% and efficacy against hospitalization was 96.4%. However, the appraisal note reminds us that year-by-year estimates can fluctuate. The later months of the trial overlapped with the COVID-19 pandemic, and a booster phase meant not all participants had identical follow-up. Next, we will turn to the crucial topic of safety. This table summarizes the key safety findings from the trial. Over 4.5 years, the incidence of any serious adverse event, or SAE, was actually lower in the TAK 003 group at 5.0% compared to 5.9% in the placebo group. While there were 11 deaths in the vaccine arm and 6 in the placebo arm, this was proportional to the 2-to-1 randomization. And importantly, none were assessed as vaccine-related. As the boxes at the bottom state, these data support a favorable overall safety signal. However, a trial of this size cannot completely exclude the risk of very rare events like vaccine-associated enhanced disease, which will require ongoing post-Lensure surveillance. Let's now look at the underlying immune response. These graphs show the immunogenicity data, specifically the geometric mean titers of neutralizing antibodies. you can clearly see that antibody levels remained higher in the TAK 003 groups compared to placebo through month 51 for both seropositive and seronegative participants. The response to DENV2 was particularly high and sustained, which is expected since the vaccine has a DENV2 backbone. The appraisal note provides crucial context. While these immunogenicity results support the vaccine's biological plausibility, they do not perfectly predict clinical protection against every serotype. Ultimately, the clinical efficacy data remains the most decisive outcome for judging the vaccine's value. We will now synthesize these observations into a formal risk of bias assessment. This table presents a risk of bias 2 style judgment for the trial's key outcomes. For overall virologically confirmed dengue, the risk of bias is rated as low across most domains. This is due to the strong randomization process and objective RT-PCR outcome measurement. For the hospitalized and severe VCD outcomes, there are some concerns. As the Y column explains, this is because hospitalization thresholds varied by country. The final 18 months of the trial were affected by the pandemic, and many subgroup analyses were exploratory. The overall conclusion is that the main efficacy finding is robust, but the claims about severity and specific subgroups are less certain. Next, we'll highlight the trial's most significant methodological strengths. The internal validity of this trial is supported by several key strengths, which are summarized here. It was a properly randomized trial with allocation concealment. It was blinded, with participants, parents, and investigators unaware of the treatment assignments. It used an objective RT-PCR confirmation for the primary endpoint, which avoids subjective judgment. Finally, it featured a long 4.5-year follow-up period. As the bullet points indicate, the large multi-country sample and baseline serostatus assessment were also critical strengths. These design choices work together to reduce bias and increase our confidence in the broad efficacy signal. However, it's equally important to acknowledge the trial's limitations. While the trial was strong overall, we must slow down our interpretation when considering several concerns. Many of the 4.5-year analyses, particularly those breaking down results by serotype, serostatus, and severity, were exploratory. This leads to issues of multiplicity, where multiple comparisons increase the chance of finding a spurious result. Furthermore, some key subgroups like DENV4 and severe dengue cases had too few events for reliable estimates. Other concerns include the varying hospitalization practices across countries, the overlap with the COVID-19 pandemic, and the fact that the study was industry sponsored. As the red bar states, these concerns weaken the fine-grained claims more than the main overall efficacy finding. Let's now consider the external validity of these results. This table addresses external validity, or where we can apply these findings. The results are most directly supported for the trial's population, children and adolescents in endemic, high-burden areas. Applicability to other groups is less certain. For children under 6, the evidence is weak. For adults and the elderly, there is no direct evidence as they were not enrolled. Similarly, pregnant or immunocompromised persons were excluded. The vaccine's utility in low or moderate transmission settings is uncertain, largely because the DENV3 and DENV4 seronegative findings matter more in those contexts. The policy lens at the bottom reflects this, with current WHO guidance recommending use in high transmission settings for ages 6 to 16. This also raises important ethical considerations. Beyond the statistics, we must consider the ethical and public health relevance. On ethics, clinical equipoise existed when the trial began, and proper consent and safety oversight were in place for this vulnerable pediatric population. Regarding equity and generalizability, the multi-country design was a strength, but the trial enrolled healthy participants, so we have less information for those with comorbidities. The most critical point is the public health trade-off, summarized at the bottom. In high-burden areas, a vaccine that reduces hospitalization is a major benefit for healthcare systems. However, using it in low-transmission settings might expose more seronegative individuals to the uncertain protection against DENV3 3, and DENV. 4. Let's clarify what this study definitively proves and what it does not. This slide offers a plain language summary of what we can and cannot conclude from this study. On the left are the reasonable conclusions. TAK-003 reduces overall symptomatic and hospitalized dengue in 4 to 16-year-olds in endemic settings, and its safety profile appears favorable over 4.5 years. On the right, we see what was not established. The trial does not show uniform protection against every serotype in dengue naive individuals. It does not completely settle the rare risk of severe disease by serotype or serostatus, and it does not prove safety in other groups, like adults, or justify use in low transmission settings. The clinical framing is clear. This is a useful tool, but not a standalone solution for dengue control. Let's now summarize our certainty in these claims. Using a grade style framework, we can summarize our certainty in the major claims. Our certainty is high to moderate high for the overall reduction in VCD thanks to the large randomized trial design and objective outcome. For the reduction in hospitalized VCD, our certainty is moderate high, reflecting the large effect but also the heterogeneity in admission practices. As we move to subgroups, our certainty decreases. For all serotype protection in seropositive individuals, it's moderate. For seronegative protection against DENV1 and DENV2, it's also moderate, but with lower precision. Finally, our certainty is low to very low for seronegative protection against DENV3 and DENV4, and low for claims about rare severe safety events. This points to what is needed to strengthen the evidence base. To build on this trial, several research and surveillance priorities are clear. For post-Lensure effectiveness, we need large real-world studies in high-burden settings to get estimates by age, region, and circulating serotypes for safety monitoring. Active surveillance for any potential vaccine-associated enhanced disease is essential. As you can see under evidence gaps, we still have questions about the value and timing of booster doses, effectiveness in adults and those with comorbidities, and performance in low transmission settings. We also need cost-effectiveness analysis that integrate vaccination with existing vector control programs. This brings us to our final synthesis of the evidence. In synthesizing this critical appraisal, we arrive at a few key messages. First, this trial should be taken seriously. It is a well-conducted, large, blinded, multi-country study with long-term follow-up and objective confirmation of dengue cases. Second, we must not overread the results. The evidence is weakest in the clinically important subgroup of dengue naive recipients, particularly for DENV3 and DENV4. This leads to the conclusion that the most defensible use of the vaccine is for children and adolescents in high dengue transmission settings as part of an integrated control strategy. The clinical message is to expect a significant reduction in symptomatic and hospitalized disease, but not complete sterilizing immunity or equal protection against all serotypes. We will now conclude by reviewing the key sources used. The information presented today was drawn from the key sources listed on this slide. The primary evidence comes from the trial itself, published in the Lancet Global Health, along with its extensive supplementary appendix. The framework for this critical appraisal and the policy context were informed by established guidelines and tools, including COSORT for reporting, Cochrane's ROB2 tool for assessing bias, and recent position papers and guidance from the World Health Organization. This rigorous foundation allows us to move forward with confidence in our interpretation of the evidence. Therefore, when considering this vaccine, always appraise the evidence, consider the context, and apply it judiciously.